Hello, everybody. Hang on, let me start sharing my screen first. Okay. Hopefully you can see my presentation. All right. So, hello, everybody. Welcome to my presentation for ESMARConf 2025. My name is Wolfgang Viechtbauer from Maastricht University, and I will be talking to you about how to visualize the amount of heterogeneity in forest plots. So forest plots started to appear in meta-analyses in the early 1980s, but these days they are really the most commonly used visualization in meta-analyses. Why is that? Well, they provide information about the findings of the individual studies and also the results of the meta-analysis itself, and they are recommended in various reporting guidelines like PRISMA, MOOS, the journal article reporting standards of the APA. Also, Cochran and Campbell both recommend or even require that the results of a meta-analysis are reported by means of a forest plot. So what does a forest plot actually contain? Well, they are a combination of textual information and various graphical elements. We start with the study identifier, typically the first author and the publication year of the study is given. Then sometimes the summary statistics needed to compute the effect sizes are provided, like mean, standard deviations, and group sizes. These allow you to compute things like mean differences or standardized mean differences, or counts, for example, for the cells in 2x2 tables to compute measures like odds ratios and risk ratios. Then the actual effect sizes of the studies are shown with corresponding confidence intervals. These are typically given as points or boxes with horizontal lines going through these points or boxes to indicate the confidence intervals. And the actual values are often also added textually, so you can read off the numbers. Sometimes the weights given to the individual studies are included in a forest plot. And then at the bottom, we have a four-sided polygon, or sometimes called a diamond. And this indicates the pooled estimate and its confidence interval. Sometimes also information about the statistical significance of the pooled estimate is provided, and also information about potential heterogeneity in the true effects. So the Q-test, the I-squared statistic, the estimated variance in the true effects. And optionally, you can also include risk of bias information about the individual studies in a forest plot. So let's take a look at an actual forest plot. This is a forest plot from the infamous BCG vaccine meta-analysis. The BCG vaccine is a vaccine against tuberculosis. And we have 13 studies here that report the number of individuals in the vaccinated group that contracted TB or that did not, and the same thing for the control group. Then here we are shown the actual effect sizes. These are risk ratios with corresponding confidence intervals. The numbers are also added here on the right-hand side. And at the bottom, we have the summary polygon or the diamond. The center corresponds to the estimated average risk ratio, indicating that vaccinated individuals have, on average, a 50% lower infection risk than non-vaccinated individuals. And the ends here of the diamond correspond to the confidence interval for this estimate. Then we have information about the statistical significance of the overall effect and information about heterogeneity. And I want to draw your attention to this because reporting this information is very important. Not only do we need to know whether a particular treatment is effective on average, this is what is being indicated here, but we also need to know something about whether the effectiveness of this treatment or this vaccine might vary across studies. And this information here gives us a clue as to whether this is the case. But this information that is typically reported about heterogeneity is, shall we say, less than ideal. The Q-test only indicates whether we can reject the null hypothesis that there is no variance in the true effects, that the variance is zero. That's interesting to know or important to know, but it doesn't tell us how much heterogeneity there actually is. The I-square statistic, which has become very popular these days, and I think we will be hearing more about this later on by a talk from Julian Higgins. The I-square statistic, what does it provide us with? Well, it tells us how much out of the total variability in the observed effects is due to heterogeneity. This can be given as a percentage, which is easy to interpret, but it's not an absolute measure of heterogeneity. So if the variance in the true effects, if we hold it constant, but we're talking about a bunch of very large or very small studies, the I-square statistic would change. So it does not tell us in absolute terms how much heterogeneity there is. Now, to get this, we could report the estimated variance in the true effects. This is an absolute measure, but it is kind of hard to interpret. So instead, or in addition, we may want to report a prediction interval. A 95% prediction interval is a range that tells us where most of the true effects of similar or new studies are expected to fall, and this directly reflects the degree of heterogeneity in effect size units. And it's quite easy to compute. We take the estimated average effect plus and minus 1.96 times the estimated standard deviation in the true effects. Now, there are various refinements on this equation to account for uncertainty in these estimates, but the principle remains the same. So this gives us a range telling us sort of how small or how large the effect might be across many studies. And this has been recommended as the best indicator of heterogeneity by various authors. Now, if we compute this here for the BCG vaccine meta-analysis, we get this range here, 0.15 to 1.55, telling us that on the one extreme, there might be studies where vaccinated individuals have an 85% lower infection risk. And there may be studies on the other extreme where vaccinated individuals have a 55% higher infection risk. So this is a quite wide range of effects that we might see across studies, indicating a quite substantial amount of heterogeneity. This raises the question of how we could show this kind of prediction interval in a forest plot, and various proposals have been made for this, and I will illustrate these in a second, and I also want to discuss two somewhat novel approaches. So the first idea is to just use the same shape that we use for the summary estimate, also for the prediction interval. Now we might want to distinguish this polygon or diamond from the one for the pooled estimate by color. But it's still easy to confuse the prediction interval with the one, the polygon for the prediction interval with the one for the summary estimate. Now, instead, we could use a horizontal line that goes through the polygon for the summary estimate, but this is now easy to confuse with the confidence intervals for the individual studies. And maybe this is a bit nitpicking, but we have to give the textual information here in a different line than where the prediction interval is shown visually. So a different idea is to use a shape that is clearly distinguishable from other elements in the forest plot, and this has been suggested, namely by using this kind of horizontal bar, and this indicates then the prediction interval. But there is a problem with this as well, namely this might suggest that every value in the prediction interval is equally likely, but this is not actually the case. So to sort of indicate this, we could use shading. Now, this has not been suggested for prediction intervals before, but Jackson suggested this for indicating confidence intervals, and we can use the same idea here for prediction intervals. So the shading indicates which values are more or less likely in the prediction interval. So darker shades of gray here, more likely, lighter shades of gray, less likely. Now, the problem with this is that it might not be clear immediately what this shading indicates, and we would technically need a legend to be able to interpret what these shades of gray correspond to. So my proposal is to actually show the entire predictive distribution, because this is what we are actually estimating. We are estimating a distribution of true effects. And here you can see that predictive distribution, and we can use optionally, but quite nicely, shading to indicate certain regions of interest. So for example, these darker shades of gray correspond to 2.5% in the lower and upper tail, and that is the prediction interval. And this lighter shade of gray here corresponds to the area opposite inside to the estimated average effect. And this area here is the probability that the vaccine is not effective, which is around 11% here. Now, this type of visualization can also be used to illustrate differences in heterogeneity across subgroups or along some kind of continuum for some kind of continuous predictor or different outcomes. So let me illustrate this. Here I separated the studies that did not versus that did use random assignment to the vaccine versus control condition. And then I used two random effects models, one for the studies that did not or that did use random allocation. And we can also see how the amount of heterogeneity differs across these two subgroups. And we see slightly more heterogeneity in this subgroup. Now, for a different meta-analysis, Banger, Drowns, and colleagues did a meta-analysis on the effectiveness of a particular intervention for improving learning. Here, the effect sizes are given as standardized mean differences. And these studies differed in size. We have quite small study to very big studies and everything in between. And I used the location scale model to not only see how the estimated pooled effect differs along this continuum of sample size, but also how the amount of heterogeneity might depend on the size of the studies. So here you see a forest plot, not showing all of the studies because it would get very, very large, the forest plot. So I'm showing you a bunch of small studies, medium-sized studies, and some very large studies. And then based on this location scale model, I estimated what is the pooled effect for small studies with a sample size of 20, or medium-sized studies, sample size 100, or large 500. And we can see how the pooled effect differs based on sample size, but also how the amount of heterogeneity depends on sample size. And we see quite large heterogeneity for small studies, and the larger studies, the amount of heterogeneity estimated there is quite small. Now, in another situation, this kind of visualization is very useful, namely when we are doing a multivariate meta-analysis, when we are meta-analyzing multiple outcomes simultaneously. And for this, I will use the meta-analysis by Berkey and colleagues, who examined the effectiveness of surgical versus non-surgical treatment for periodontal disease. And two different outcomes are typically reported in these studies, namely the so-called probing depth and the attachment level. Without going into further details what these outcomes represent, here you can see the 5 studies that were included in this meta-analysis and the effect sizes for these 2 different outcomes. And then using a multivariate meta-analysis, we can estimate what is the pooled effect for this outcome versus this outcome. And we can also allow the amount of heterogeneity to differ for these different outcomes. And we see that there's more heterogeneity for the outcome attachment level compared to probing depth. Now, all of the models I have used so far assume that the true effects are normally distributed, but this may not be the case. We can either assume a different distribution or estimate the distribution of true effects non-parametrically. But we can use the same visualization when using these methods. Now, I should note that other visualizations would hide the shape of the distribution of true effects, except when using shading, but this is difficult to process. So here you can see the same forest plot for the BCG vaccine meta-analysis and the predictive distribution assuming normality. And then I use this non-parametric method to estimate the distribution of true effects. And this looks quite different. And here you can see that it's a sort of bimodal distribution. So we can see exactly what that shape of the distribution looks like. So to conclude, the predictive distribution is clearly visually distinct from other elements in the forest plot. It clarifies that we are estimating an entire distribution of effects. It visualizes that the values under that predictive distribution have different likelihoods, and we can use shading to emphasize different regions. It is very easy to create these kind of visualizations using the metafor package, and you can find code corresponding to the examples shown in this presentation on my website. So those are the references, and that's it for my presentation. Thank you for watching, and I'm happy to take any questions, comment, or suggestions.
Thank you, Wolfgang. I'm just checking the chat. I don't see any questions yet, but just give it a moment because we might be a bit behind on the stream. Okay, we've got one from Matt. Looks like a nice addition to a great package. Will you be extending this to confidence intervals, or is there a reason not to?
So while one could show also a distribution for the confidence interval, that would be also a possibility, but I would be a bit afraid that the forest plot gets very cluttered when doing this. Now, this idea of using shading for confidence intervals, that could be another possibility. So that's something that Jackson suggested. And I might be adding this at some point in the future, although I think people are very used to seeing these boxes and the lines for the confidence interval. So not sure how much that would catch on, but it's definitely an interesting idea.
Thank you. Sorry, I'm just waiting for the YouTube to catch up. Samuel says, thank you for this interesting presentation. Do you see challenges in communicating predictive distributions to non-statistical audiences?
Well, so it could be that people are initially a bit surprised. What does this distribution signify? Although this idea of showing this normal distribution has been used in sort of some tutorials on teaching about random effects models, or also Borenstein and colleagues in their book on Introduction to Meta-Analysis, they kind of show that normal distribution to show, well, we are really estimating a distribution of true effects. So it's not a completely novel idea. Um, but I think it's actually quite important to kind of emphasize this point because that is really what we are doing. And so I think it's just on us to clearly communicate what does this represent, and in teaching about meta-analysis, that this is what we are doing. We are assuming that distribution, or we can estimate it non-parametrically. And so I think that's the challenges on people teaching about meta-analysis, what this really represents.
Okay, thank you. I think that's all for questions. We do have a comment that says interesting idea, and Matt says, yeah, makes sense. Thanks for your previous question. Okay. Yep, we've got another comment just saying, very good presentation, Wolfgang. Thank you. Okay, so we'll move on to Lena.
Hello, I hope you can all see me. And let me actually share my screen now. I hope you can all see my screen. Can you all see my screen?
Yes, we can currently see your PowerPoint window.
Oh yeah, okay, perfect. I'll get started then. Hello, so my presentation today is about why do we need to randomize and split data, and part of the motivation of doing this presentation is actually that we did a tool to help people doing that during the evidence synthesis hackathon, which is a hackathon that the attendees are probably going to be familiar with. Because it's the one that's organized by Matt and Neil. And basically, we coded a tool called Ref Randomizer to help people with set data splitting. So briefly to introduce myself, I'm Lena. I'm a PhD student at the NIHR Innovation Observatory in Newcastle University in the UK, and I'm interested in automation of systematic reviews and automation of data extraction. And I've also recently started a position at UCL, and my main sort of research interest there is living reviews and climate and health research. And at the 2025 Evidence Synthesis Hackathon at UCL, as I said, we created Ref Randomizer, and the people that contributed to that are Fiona Campbell, who's also from Newcastle University, and then Matt and Neil, who actually originally proposed the project. So to get started, why do we need to randomize? So this is just a familiar example. So many of those of you, of course, who are listening are, of course, very familiar with systematic reviews, and especially the systematic reviews where we're looking at effectiveness and treatments. We use randomized controlled trials, so randomized trials. And one of the main reasons why we use randomized trials is because of the randomization being good for the validity of results. So it reduces the risk of having biased groups. So, for example, baseline differences between the groups are skewing the results through selection. It's of course also important for statistical analysis and ethical considerations. What I'm talking about now is slightly different. So I'm talking about randomizing references. So if we search references, for example, from PubMed, they will be downloaded, and quite often they are ordered in some way. So that might just be like a subjective ordering by PubMed, putting what it thinks is most relevant to a query on top, but it might also be a date or an alphabetic order. And that means that if we take a non-random selection of our records to explore a data set or to train an AI, our data set will not be representative of the whole set of records. So for example, this could be when there's lots of authors whose name starts with L or something like that. And if we just take the first couple of records from a data set, we might just end up predominantly, for example, for papers with authors from a certain region. And why would we want to randomize references? So firstly, if we want to test agreement between reviewers in a systematic review task, so for example, if we want to do some pilot screening, where we want to obtain random sample of references for people to dual screen, to get people on the same level, or also to test some data extraction. But then also, maybe more importantly, when we're training, developing, or testing AI-related methods to automate reviews, we always want to split our data sets randomly as well. And one of the examples or analogies to explain the importance of this is, for example, students taking exam. So first, student would study material to prepare for an exam. So they train themselves. And then they might take a mock exam or a practice exam where they're using new but slightly different materials and questions to sort of see whether they get what they want to study for, or whether they need to go back and do another training step, and whether they need to learn for. So this is like a sort of circle process. And then lastly, when if the students feel that they're confident, they can take the final exam, but they only have one chance to do this. So these questions should of course be different to the previous material to show to see that the student actually understands and is able to apply and transfer their knowledge. This is the testing stage. And the AI use case is similar. So we do also quite often need these 2 or 3 different set of records to train, test, and validate robust models. And so there are actually quite significant risks when we're developing AI if we're not randomizing our references. Because if the AI will be really good at answering a question, for example, making a screening decision or extracting a certain data point to which it has already seen the answer in the training process. So that there's this problem of data leakage. So we test the model on the data that it's already seen, then we will overestimate the performance of the model, and the model will be unsafe to use downstream. And then overfitting, and that's just if we don't randomly shuffle our data before taking the selections, we might just fit the model overly to data with one specific characteristic, and then when in downstream prediction the model sees slightly different data, it might just act sort of erratically, and it might just act unpredictably. So yeah, we need to prevent data leakage because we do not want to overestimate the performance of our model. So while it's great that if we have a screening algorithm, screening automation tool that gives us 99% sensitivity on a certain, selected test set, if we then apply the same screening model to a downstream set, and actually it was overfitted or there was data leakage and it will only identify 70% of our records, it would be very hard for us to figure that out at that point. So it's very dangerous. So we need to make sure that, we split data sets, because data leakage happens if we don't split the data sets. If we use a non-deduplicated data set, so that means that if we, for example, retrieve references from PubMed and from Scopus, and we don't deduplicate them against each other, and then we split them into different data sets, might mean that the same record is in the training and the validation data. So we need to always be careful to deduplicate. And also, because there are many already labeled gold standard data sets out there for AI training, you also need to make sure that those are deduplicated against each other, because quite often they just take portions from each other. So one gold standard data set might just take some data points from a previously published data set, and if it's not clear, then you will end up with some data leakage, or of course a splitting and randomization tool might glitch. And so sort of a practical example that might be familiar to those of you who are interested in looking at automations through LLMs is that LLMs have a slightly different process to this training, validation, and testing process, but they have a process of prompt development. And that applies to if you're using the LLM for screening or for data extraction. So first of all, you should always use a random subset of records to develop and adjust prompts. So that's the smallest sort of subset of records that you need. So that is, you're just running the LLM over and over again on the small data set of, say, 20 records. And then you're looking that the prompt that you're developing should perform sort of perfectly on these small records. For example, you have 20 records, and that's an independent data set. And then you need a second independent data set, which is a testing set, where you apply the same prompt that you've developed in step 1. You apply it on step number 2 to see how well the LLM actually does. So at that stage, it's really important that you don't adjust your prompt again, because then you would overfit to this independent data set. And this is usually a slightly bigger data set, like say, for example, 200 records. And then you have to compute the same validation scores again, and you can often see a drop. So you can see that the data set where you fitted your prompt to, the results will be really good, but it might be slightly worse on the second data set. And only if you have like a robust evaluation on the second data set, then you should sort of consider applying your LLM on downstream data that doesn't have any labels yet. And then if you're reporting what you've done with the LLM prompt development and automation, it's really important that you report that you did the random data set splitting and that you also show your group sizes. So here the 20 and the 200. And, you know, in randomized controlled trials, there is a lot of guidance to say that group sizes and, you know, the method of randomization have to be reported, and just for the same reason, we should report it here as well. I think recently there's been a little bit of a trend in some of the automation publications where people, I think, unknowingly didn't report it, or they didn't do it properly because they weren't aware of it. So it's really, really important that we report things correctly. And actually, if you have the link to my presentation later, there's actually a reporting template that I've linked here, especially for LLM and systematic review automation. And so what we've done here is we've created Ref Randomizer, and Ref Randomizer is a way to help you to split RAS or CSV files in any number of independent data sets. So you can upload the RAS or CSV file, but if you do upload an RAS file, it'll actually return both RAS and CSV. And the reason why that is quite helpful is that for LLM processing and, you know, feeding articles in the loop to an LLM, actually CSV, if you're interacting with APIs, is slightly more easy. And yeah, you can literally split any data set, so it doesn't even have to be references. It can be any sort of data points, as long as it's in a CSV file. You can upload it to the tool and get your data splitted. And let me see, I will now just briefly share the tool with you, and please somebody unmute themselves and tell me if you can't see my screen right now, because I've just switched to the browser. So this is reference.
Yeah, we can see your screen.
So you can see the Ref Randomizer now.
The
web app. Okay, cool.
Yep.
Thank you so much. Yes, so this is deployed. The link is in the presentation as well. And so basically what happens here is you can upload some files. So here I'm going to upload an RAS file. And it's going to tell you how many records were in your RAS file. And now this is here where you can define your record batches. So if we're sort of hypothetically thinking that we want to develop and validate and sort of apply an LLM on this, we first need our prompt development data set. And here you can give any percentage, and then this will be the percentage of your first batch. So if I say my first batch should be 5%, I can say 5 and enter, and the tool is going to tell us that 5% is going to approximately be 37 records. And you can see that 5% of our data set has now been allocated, and 95% is left. So if we now want a slightly bigger validation set, so say 15%, we can give the 15% here, and now it tells us that this would be 113 records and 80% is left. So if the 80%, that will just be the data that I'm going to run the model on downstream. You can see 100% has been allocated, so you can submit it here, and you can download a zip file. And then if you open the zip file, basically what you're going to see is you're going to see your batch data here. And so anything that comes in the CSV will be in this format. And then you'll also have an RAS file though. So batch 0, and you can see the sort of size of the batches as well, sort of written into the file name. So you know that this is the smallest batch, and then the medium batch and the big batch. And oh, now I can only start from the beginning. So I'm back in my presentation. Sorry about that. I should have just to briefly finish. So yeah, so this is the overview of the tool. And yeah, as I said, this is the evidence synthesis hackathon open source tool. If you scan here, you'll be able to find the tool. But then also there's a GitHub. So the tool or the interface that you've just seen is actually Python and Streamlit coded. But it's available in the evidence synthesis hackathon repository. And there was some original R code as well from Matt, I think. So that is currently in a branch. So if you you're looking at, some of the, I think, R functions, or if you want to sort of develop like an R counterpart for this tool, there should already be some sort of useful code in there in the R branch. But then, yeah, the deployed web app is here. So anytime that you want to split or randomize data, you can go there and use it. And we've also on the tool website and on the OSF published a sort of information sheet. So this is just a white paper, but this also gives some more background on sort of the methods and the ideas behind the tool. Here are the references. Yeah, and that is it from my side.
Thank you. I'll just give the stream a minute to catch up. While we're waiting, I will take the ability to ask you a question. What prompted the shift from R to Python?
Well, so this would be entirely up to me because I quite like the suggestion of the hackathon project, but I'm just not a R native. So for me, I've done just lots of Streamlit apps and Python apps, and Streamlit is exactly the same as Shiny, but in Python. So yeah, this would just be down to me signing up for the project. And I think the evidence synthesis hackathon also, that there are a few Python people participating there these days as well. So yeah, that's the main reason.
Thank you. I hope that wasn't a difficult question. We do have one from Louise. Is there a way to control the randomization process, e.g. using a seed?
Yeah, I think there should be a way to implement that. I remember when I was testing the tool and when I was developing it, I think sometimes when the tool is deployed, it seemed like the random seed wasn't quite working, and I wasn't sure why, but I didn't get much time to investigate it. But I think might be because of the way how the tool is deployed. But yeah, I think there is an option to add the random seed in there as well.
Thank you. Trevor has said, looks great. Thanks for sharing.
Thank you.
I believe we might have one more. Give me a second. I'm just... Wolfgang has asked, does it automatically check for duplicates in the references uploaded?
No, it doesn't do that. I think it could potentially do that with like exact matches, so exact matches within a column. But I think the thing is the deduplication, I think, is quite a complex process because sometimes duplicates don't have to be exact title matches, or there can just be a copyright statement in the abstract that will be different, that will then prevent the abstract to be a direct match. So I think deduplication is such a sensitive issue that it needs, I think, dedicated tools in order to do that. So that's why I've sort of steered clear from it for now, just because I wasn't able to properly implement and validate it. But there are, I think, tools that'll help with that.
Yes, I believe we have some ES hackathon tools. I know at least Citation Chaser should deal with that. I don't know if it's the best tool in this instance, but there are tools available. I think that's all for questions. So thank you very much to our two presenters. We will now be going back to YouTube onto the playlist for more presentations. Thank you very much.